Clear the back corner.
Copy, clear back corner.
I've got, yeah, good to start mezzo. Yeah. Okay. Roger, would you like all the front ones on? And also your aft. We'll leave your port starboard off for now.
Control winch.
Go for winch.
Yeah, hey, are you guys getting a speed readout on the winch on your guys' GUI?
I am not. I also
We are not.
don't have a...
Okay, we'll stand by once we get... Yeah, I got nothing as far as speed or meters out.
Yeah, same.
Same with us, we are not getting any input.
Okay, nice. So DC ground fault on Atalanta's down at 4 megs. I can turn SVS off for now, yeah. Hope it squeeze out.
This is an audio slate for dive H2109. UTC time is 020330.
Control winch. Will you let me know when depth for Atalanta has reached 50 meters?
Copy, we'll let you know.
I'm at 25 meters. 25 meters currently, yeah. Video, I'm turning on some lights on Atalanta, so that might mess with your video values. That's about half our lights on. It's just not wire out, and yeah. I'm gonna turn your optode off again because I'm still tracking down this slight DC ground fault. If thank you. Nice. He's at 50. I'm at 47. Yeah, I'll let you know. All right, 50.
Winch, this is Nav. We are at 50 meters.
After that, I'll stop 50 meters and transferring remote to the van.
Copy all.
Yeah, that's awesome. Are they ready for me to bump the winch out a
Control
bit?
winch, go ahead and give it a small bump.
There we go.
Copy.
Okay, got you.
All right, I've got control. Okay, so update for you. We've got a small DC ground fault on the optode. It's not super low. It's like 200 kilo ohms right now. We can leave it off or we can keep it on. And we can leave it off and hope it squeezes out, maybe. Okay. SVS is also off on Atalanta for now. We're waiting for that to squeeze out. Gauges. I don't know. That is maybe something Robert should be consulted on. Robert and Deck should be talking about that, yeah.
I think I can go in NAS. Hi, Rye and Levi. Would now be a good time to kind of go over some introductions and talk about what we're doing for this dive?
You're really quiet as well. Sorry.
Hey Ray, we'll get some levels for everyone as we do introductions.
Cool. I think, Matt, you're asking if we're ready to do intros? I think we would like to hold off a little bit.
Sounds great. Thank you so much.
Thanks. Okay, gauges look good. Um,
check levels for SPL and records. That's all looking good. All right.
Yeah, are you talking on SPL?
My bad, I'm here now.
Okay, cool. It's like I feel like I can barely hear you. Yeah.
All right, so hi everyone, my name is Matt Holden. I am the Science Communication Fellow right now for this shift on NA174 as we explore deep sea habitats around the waters of the Republic of the Marshall Islands. We are so excited to be here today. This is our first dive of the expedition. We just got our ROVs into the water. They're currently descending down to 2,200 meters depth as we search for new species and range extensions. We'll also be looking into some geology and collecting rock samples to analyze for age dating. And one of the really cool things I'm excited about is we'll be collecting some eDNA samples using amazing new Multipuffer as well as Niskin bottles. So I just wanted to go around and give a little introduction to everyone on the team in the control van right now. As I said, my name is Matt Holden. I'm the Science Communication Fellow for this watch. When I'm not out at sea, I am a high school science teacher at a public high school in Fayetteville, Arkansas, and I'm just so happy to be here with you all today as we go through this exploration. If we want to go ahead and start with introductions, Annette, would you mind telling us a little bit about yourself, and then we can kind of just go clockwise around the control van?
Hello, can you hear me now? Perfect.
Yes, awesome.
Okay, thank you. I'm Annette Govindarajan. I'm a biologist at the Woods Hole Oceanographic Institution, a co-lead on the cruise, and I'm doing eDNA sampling with the new Multipuffer sampler. It's a large volume eDNA sampler that filters the water in situ, and it's produced by Aquatic Labs in Cambridge, Massachusetts, and we are trying it out, this new model, for our first time. Thanks.
Awesome. Thank you so much, Annette. This is so exciting to get to try this new technology for the first time. I
Yes.
know that I'm just ecstatic to see what we find.
Yep.
Kelsey, would you mind telling, Dan, would you mind telling us a little bit about yourself?
Yeah, absolutely. I'm Dan Kirlov. I'm our video engineer. This is my first time out on our dive, and I'm really excited to see what we find today.
Awesome. Thanks so much, Dan. And if we could get our ROV pilots, Rye, would you mind telling us a little bit about yourself?
Sorry, is it my turn?
It is.
What am I supposed to say?
Just a little bit about yourself.
Okay.
Maybe, is this your first time, or have you done this before?
Yeah, I've been coming out on the Nautilus as ROV team since 2023, I think. And my name is Rye, and I'm in the co-pilot seat today. And anything else?
That sounds great. Thank you so much, Rye. We are so happy to have your amazing experience in this dive. Levi, could you tell us a little bit?
My name is Levi Yinema, and I'm sitting as Hercules pilot right now. And we are descending. Looks like ROV depth is 313 meters. We're going down. I am from Newport, Rhode Island. This is my first cruise flying Hercules, so very exciting.
Yeah.
Thanks for having me.
So excited. Gabby, would you mind telling us a little bit about yourself?
Yeah, of course. My name is Gabby Espino. I'm the navigator for this cruise, also seafloor mapper. I am originally from Kailua, Hawaii. I am a Navy veteran. And yeah, just graduated from University of Rhode Island online program. So shout out to URI.
Woo woo.
Great people there. And yeah, I'm excited to be diving.
Yeah, we are so excited to have you, Gabby, and congratulations on your recent graduation. That's amazing.
Thanks. Yeah.
Awesome. Leela, would you mind telling us a little bit about yourself?
Hey y'all, I'm Leela Madhava. I am in the data logger position, meaning I'm keeping a record of important events as they happen during the dive. I'm originally from Chicago, but I'm currently studying marine biology at University of Washington, Seattle. And this is mostly my first time out to sea, and I'm super excited about it.
Awesome. We're so excited to have you, Leela. And Mariana, would you mind telling us a little bit about yourself?
Mariana, and I'm on the Science 2 position, and I'm from here in the Marshall Islands, from Mimra, and yeah.
That's awesome.
Yeah.
We're so excited to have
Hi.
you and get to learn some expertise from you as someone from the Marshall Islands. It's just such an amazing opportunity to have you on board. And then Steve, our lead scientist for this dive and expedition, would you tell us a little bit about yourself?
Sure. So my name is Steve Oskovich. I'm the watch lead for the 12 to 4 and one of the co-lead scientists for this expedition. I am a biologist based at the National Museum of Natural History in the Smithsonian Institution in Washington. I am a biologist who focuses on the genomics of deep sea organisms and a little bit of environmental DNA and how we can use new genomic tools to better explore the ocean.
That's awesome. Thank you so much, Steve. Like I said earlier, I'm just so excited about the eDNA samples we're going to be collecting and what we're going to find. As a biology teacher, I'm always trying to teach my students about new and innovative technologies to explore the world, and so this just means a whole lot for me personally. So those of you for just joining, we are on SPL, which stands for Science Party Line. This basically allows everyone in the control van, from our videographers to our data loggers, scientists, our navigators, ROV pilots, all to communicate to each other and then also share what we are experiencing with you all around the world. So we're so happy to have you here as we embark on this amazing expedition. So I did have a question come in about the weight of ROV Hercules. Would one of our ROV pilots maybe mind explaining a little bit more about our ROVs that we're using on this dive?
Sorry, Matt. ROV's a bit occupied right now. Maybe come back to us.
Of course, sorry about that.
No, it's all good.
So, as I mentioned earlier, some of the main objectives of this specific dive, we are just east of Eiling Laplak in the waters of the Republic of the Marshall Islands. And I was wondering, Steve, could you tell us a little bit more about our science objectives for this dive?
Sure. So we're in full exploration mode for this expedition. So that means basically we're going down to the seafloor in areas that we haven't yet explored or even mapped. In fact, this feature was unknown to us in its shape and size until just earlier this morning, about 6 hours ago. We were able to get some good first maps, high-resolution maps of this seamount. And our goal here is to explore the deep sea benthos, so the animals and habitats and ecosystems on the seamount landscape, and also to understand some of the structure that forms the geology of this region. So we'll be looking at the shape of the seafloor, the things that live on the seafloor, and trying to compare it to what we might know about areas adjacent, for example. We've tried to put all of our sites in context. We know that every ecosystem has the potential, every deep sea environment has the potential to host new and novel species, but we haven't been here before, so we're going to try and compare it to something that we might know a little bit about, and hopefully that'll help us tell how unique this environment is and how unique the animals that live here might be.
That's great. Thank you so much, Steve. Yeah, it's just so exciting to get to explore these areas that have so little scientific research in the past done here, and getting to kind of see firsthand, be some of the first people to explore these amazing ecosystems.
Yeah.
So we are currently just around 490 meters deep as we are descending down to about 2,200 meters for this dive. Again, just east of Eiling Lap Lap in the waters of the Republic of the Marshall Islands.
Good siphonophore there.
Oh,
Huge.
wow.
We're passing through the deep scattering layer right now, kind of the core of it. And it's a part of the water column where a lot of deep water animals are found in high density. So there's some fishes, some squids, some cephalopods. These are animals that live exclusively in the mid-water column. And actually this was a feature we were able to identify in sonar because a lot of the animals that live in this area are so dense that they scatter the sound we use to map the seafloor. So you might see some siphonophores and some stringy creatures and some small critters zooming by on our descent. There you go.
That's awesome.
Yep.
So these critters that we're seeing, how do they feed? Do they have like special adaptations to allow them to feed in this type of the environment?
Yeah, a lot of them are, a couple different strategies. Some of them might have long tentacles to wait and capture things as they swim by. Some of them might be ambush predators that lay still. Some of them may be and consume prey. Some of them might have bioluminescent aspects to them that attract prey items that they can feed on. There's a lot of different adaptations for feeding in the midwater.
Super interesting. I love learning about these unique adaptations. When I teach my students, especially in our evolution unit, we talk a lot about these really cool adaptations that these species have evolved to have to allow them to thrive in these really unique environments. So I'm so excited we're getting to see it firsthand. So, Annette, would you mind telling us a little bit more about your eDNA multipuffer and kind of like how it works?
Yeah, sure. So, I've been working on deep water column sampling for several years, and we first did firstly, guess I would like to explain what eDNA is, right? I think that might be helpful.
Yeah, totally.
So for us animal biologists, eDNA refers to the trace genetic signatures that they leave behind as they move through the water. So animals can slough off cells, they have fecal pellets, there's little bits of tissue, it's little particles of themselves that get left behind. And so if we sample them, it's like we're forensic scientists trying to figure out who they are from their genetic signatures. So to collect these genetic signatures, we have to filter the water. And what we found the first time I sampled in the deep water column, the obvious striking thing was that the eDNA became very, very sparse as you went deeper. And so the typical way of collecting water for this is with the Niskin bottles. You use these bottles like Hercules has, and you collect the water, you bring it back up, and you filter it. But those bottles only contain a few liters of water, right, a piece. And so it seemed to me that we ought to be sampling much larger volumes of water. And so we that directed the development of our autonomous sampler, which could filter water in situ, so it saves some labor as well. But it could also filter large volumes, and we put it on the first versions, we put on Mesabot. So for maybe some of the listeners who've heard about some of our other Nautilus cruises are familiar with the Mesabot, which is our midwater robot. And so we've done very successful sampling on that with our what we call our oceanic HUI samplers, We're now trying out the Multipuffer, which can go deeper. And so that's what we're aiming to do as we go, below 2,000 meters today. We hope to be collecting our large volume samples along the transect and maybe hopefully see some different genetic signatures that might be associated with the change in the topography. So
That's so interesting. I mean, I feel like that multipuffer is going to give us, so much more data and allow us to really get a better overall picture of what organisms may live in these environments all the way throughout the water column. Is this something that can only be used in saltwater, or?
no, so you can sample eDNA in any type of water, freshwater. In fact, probably the beginnings of the field were in freshwater, but you don't need the same large volumes. I've actually done some sampling in river systems, specifically the Fraser River in British Columbia, and you can't get large volumes because there's so many sediments, and you're lucky if you get a liter of water, and so but that's enough to get the signal. So you have different. There's actually now a variety of different autonomous sampling tools out there, right, in the field, and different tools, different ones are suited towards different environments. There's no one size fits all, and so it's actually a really great thing that there's been a number of different efforts by different people at different institutions would come up with different solutions that are suited for different environments, because the ocean's a big place. We need a lot of solutions to this immense challenge that we have.
Yeah, very, very cool stuff. I definitely think that when I get back to Arkansas, I'm going to try and incorporate some of these eDNA sampling methods in my biology classes.
Right.
I think it'd be a really cool, authentic way for my students to learn about these amazing technologies.
And then one thing I was curious about, Annette, you mentioned the multipuffer can go deeper than the Mesobot can.
Yeah.
Do you know what is it about the way that it's built that allows it to hit those deeper depths?
Well, so we're talking about the samplers and the Mesobot. Just to clarify that the samplers are separate from Mesobot. Mesobot can go down to 1,000 meters. Those particular samples can go down to 1,000 meters. It's in fact, I've never tested them. There's pressure testing facilities, right, where you can test out materials and equipment to see if it can withstand the immense pressures, because as you go deep, right, it's crushing. What we like to do a lot of times, and maybe we'll get to do at some point on this cruise, is put little styrofoam cups out, right, and then they shrink due to the immense pressures, right, and so we have to test it out, and I don't want to risk it. I mean, those samplers may work, but I'm comfortable with using them to 1,000 meters, and we have this other, some different materials, I think, but the intent is that we'd like to use them down to 6,000 meters. We still don't have them ready to go down to the hadal depths, but maybe someday.
That makes sense. Thank you.
Yeah, thank you so much, Annette.
Sure.
It's a great explanation. So for those of you just joining us, we are again descending on our first dive for NA174 in the waters of the Republic of the Marshall Islands. Hercules, one of our ROVs, is currently right around 745 meters deep as we are descending to about 2,200 meters deep on a newly mapped seamount to basically explore the biodiversity, what species may live there. Maybe we find some new species or some range extensions. As Annette said, collecting these really cool eDNA samples that we can analyze back in the lab to get a better overall picture of what this ecosystem looks like and the organisms that live there. And then we're also going to be collecting some geology samples, some rock samples to basically date and again tell us a little bit more about the history of life on Earth.
a third of the way there.
Awesome. Getting closer. So one of our awesome science interns that we have with us on this dive is Leela. And I was wondering, Leela, if you would tell us a little bit about like how you got into this field or how you got interested in ocean exploration in general.
Yeah, sure. So I actually, when I was growing up, I didn't really have any real interest in marine biology, which I think is different from a lot of my classmates. I grew up in Chicago, so it's a lot of freshwater around. And I love the ocean, but I didn't think much about it as a career. And then when I was accepted into University of Washington and was first picking out all of my courses for my first year, like everyone that was standing out to me and I was like, oh, this looks so interesting, was marine biology related. And then actually a few weeks later, I went to one of Nautilus's ship-to-shore events and like heard y'all talking and was immediately like hooked and was like, I want to apply for this right away. So that's actually why I initially declared my marine biology major, because I was like, oh, if I change my mind, I can always undeclare it later, but you're required to be some sort of science major to apply for this. So yeah, I declared it to apply for this, and then I didn't get in the first year, and then I did get in the second year, and now here I am.
That's so great.
Yeah.
You know, one of the things that you just said about not getting in the first year, but you didn't give up. You kept trying again, and I think that's so important. You know, I know personally I've applied for many things and been rejected, but you got to keep trying. You got to keep putting yourself out there. If you're passionate about something, I think it's super important to never give up, and obviously we can see that in you too, Leela, so super amazing.
Yeah.
I guess as we're talking a little bit about kind of how we got into this field and like why we're interested in ocean exploration in the first place, Steve, would you mind telling us a little bit about how you first got started or how you first became interested in ocean exploration?
Sure. We've got an hour and a half, right?
Maybe not all the hour and a half.
All right,
Maybe
I'll give you the abridged version. All right, so for context, you know, I went to undergraduate program as a marine science major, but I was convinced I wanted to study fishes and fish ecology when I graduated, and deep sea was not on my mind at all. You'd be surprised. Anything can happen when you move out of the space of your undergraduate program and you have a lot of different opportunities to explore what you can find. So I was one of the early Nautilus ocean science interns in 2012, I think was my first application. And so I got to sail for the first time in 2013. And I was kind of hooked on that. And I was at the time also pursuing a graduate program, a master's in deep sea biology, but marine science in particular, marine biology. So you never know what path you're going to take, you know, once you leave your undergraduate program. You might have an internship, you might want to go to grad school. There's an option for everyone, but sometimes you don't know what you're going to be good at or what's going to attract your attention until you try some things. So that I was lucky I found something that, lucky and motivated that I was able to find something early that I could latch onto, and here I am in my 24th Nautilus expedition.
Wow. That's incredible. That's so cool that you started as a science intern and now you're on your 24th expedition. You're one of our co-lead scientists leading this watch. Like, what a cool, like, kind of full circle story. Really, really cool.
I'm pretty sure they only bring me back for the dad jokes, though.
We're gonna have to get some of those.
It helps keep people motivated at 2 a.m.
Do you have?
You're gonna want to hear some in about 12 hours time. Blue water, 2 a.m.
Do you have any favorites out of all those expeditions? Like anything that sticks out to you when you think back on like?
Yeah, I still think that NA110, the Line Islands and Jarvis Islands cruise. I just saw that was my first cruise as a science co-lead. And some of the things we saw were just mind-blowing. Like, working in the equatorial Pacific, I think, has always been a career highlight for me because it's such a unique place where you have a lot of biodiversity that is uncharacterized, and everything you look at could be new. And it usually is new. In fact, we have some confirmed new species from those expeditions that were recently published. And I think if I ever got the chance to go back there, I would drop everything and try to take that opportunity. Yeah.
Very cool.
Sounds so cool.
Yeah, that's just amazing. Yeah, I like how you said, almost like you never know what your experiences are going to bring next, kind of where life leads you. Not all of us got to where we are, or got to work with OET through the same path, and that definitely hits close to home. As an educator, that was not my first path. I originally went into wildlife conservation biology and then eventually transitioned into education, which eventually led me here as a science communication fellow aboard EV Nautilus. So I think it's important for our viewers to know, especially our younger viewers, that things can change throughout your life, and you never know what the next day may hold. Hey, Levi, would now be a good time to talk a little bit about Hercules?
Sure.
Yeah, so we have a few questions.
Yeah.
One of our questions is, how big is Hercules? Like, how much does it weigh?
It's about 6,000 pounds in air, but once we put it in the water, it's actually about maybe 50 or 100 pounds buoyant.
Wow.
Oh, wow.
That big foam pack is roughly half of the weight of the vehicle in air, syntactic foam, but once it goes in water, it's actually buoyant.
Very cool. And so, as it gets into the water and starts to descend, can you speed it up or slow it down, or does it have an average quickness that it normally descends or ascends?
So descent on these systems is typically 30 meters per minute, and that's kind of a limit of all these ships have a very similar winch system, but also the vertical thrusters on the ROV kind of limit the speed too. They're actually about the same. So 25 to 30 meters per minute is kind of a standard speed for ascent and descent on these type of cabled vehicles.
Very cool. And so it has thrusters that allow it to move up and down, obviously. Yep. Does it have other thrusters that kind of allow it to navigate throughout the water column?
Yeah, so it's a two-body system. So you have Atalanta, which is a stainless frame with lead on it. Think of it as a weight at the end of a steel cable. That has two thrusters for heading control. And then there's a neutral tether 30 meters long going to the free-swimming ROV Hercules. And ROV Hercules has 6 thrusters, so it can go anywhere. And that neutral tether takes out the ship's movement. It decouples the movement of the ship to the ROV camera system, basically. So as Atalanta goes up and down with the ship, with the heave, with the swell, the waves on the surface, so Atalanta goes for a ride up and down. The ROV, would think of it like a dog on the leash, is decoupled from all that movement. And then the ROV can move really smoothly and can park on the bottom.
That's awesome. I'm sure that helps with collecting, different samples of different rocks or biological samples, especially some of those that may be more sensitive or have kind of those body types that may break apart easier.
Exactly. Yeah, you need to be, for this delicate work, you need a precise movement and good control to carefully maneuver the ROV right up close to these corals, these sponges, these living animals, so that way we can videotape them and we can sample them.
Very cool, very cool. Yeah, when I first learned a little bit about kind of that decoupling system, it was explained to me that it basically that system of Atalanta and Hercules working in tandem, it basically prevents Hercules from bobbing up and down like
Yep.
a tea bag would in a cup of water. So I'm sure that that really helps with some of the more delicate maneuvers.
Exactly.
Looks like we're just about halfway there, guys.
Ooh, exciting. I can't wait to see what we find. Also, something really cool about EV Nautilus that allows us to basically be in contact with people from all around the world is our telepresence system. So this allows us to basically show what ROV Hercules is seeing, send that up to Atalanta, which sends a signal up to the ship, eventually getting sent up to satellites, and then back down to Rhode Island, where that signal is sent out across the world so that anyone with internet access can watch what we're doing, watch our operations pretty much 24/7. For example, right now we have people from the United States, Canada, Germany, Australia, Guam. So there are people kind of watching this exploration and this expedition in real time from all around the world, which I think is just super cool. So again, those of you who are just joining us, we are currently on our first dive of NA174 in the waters of the Republic of the Marshall Islands, just east of Eiling Laplap. This dive is going to take about 12 hours, give or take. And again, we are looking for unique life forms, looking at the biodiversity of the life that we might find down around 2,200 meters on this newly mapped seamount, as well as collecting eDNA samples and geologic samples to, again, tell us a little bit more about the history of life on Earth. So we just had a question come in about some unique challenges of being on expedition at sea for first timers. This is my first time at sea on an expedition. So some of the unique challenges that I've experienced would just be kind of getting used to our watch schedule. So as our regular Nautilus Live viewers know, we are broadcasting 24/7, and so all of the people, all the team members aboard EV Nautilus are kind of sectioned into different watch schedules that run for 4 hours, and we do 2 of those every 24-hour period. So kind of getting used to the sleep schedule has been a little bit of a challenge, a little interesting for me. And then just kind of being away from land, it can be a little daunting to look out for multiple days and not see anything but blue water, but it's been a really amazing opportunity and expedition. Any other first timers want to tell us a little bit about challenges on being on this expedition or being at sea for the first time?
I have one.
Yeah.
So I'm clumsy enough on land, but at sea with the movement of the ship, it does make it like 10 times worse. So I have spent most of this cruise either crashing into doorways or hitting my head on things. So there's been a lot of that.
Yeah, I definitely have bumped my head and my shins a few times walking up and down stairs. So I definitely feel that challenge along with you. I think another one would just be getting used to the movement of the boat. So we are out in, the middle of the Pacific Ocean, and sometimes the weather is not as calm as it might be today, and it can cause some pretty big swells and kind of move the boat around. And so getting used to that and, trying to prevent seasickness is definitely a real challenge for many of us, especially as first timers out here. Dan, are you available to talk a little bit?
Yeah, absolutely.
Is this your first time at sea? I can't remember.
This is my first time at sea, so, I feel like you've touched on kind of most of the points, but yeah, definitely, especially on the taller side, I don't think these ships are made for that. So I've hit my head a fair few times, and then, yeah, the swells with the seasickness and everything like that, and you just kind of learn how to move with the motion of everything, but
definitely.
I think after a few days, I think everyone gets there.
Totally, I agree. I think it's important, drink a lot of water, make sure you're hydrated, make sure you're taking like electrolyte packets and things like that,
Absolutely.
Eating regular meals. I think that also helps a lot with that seasickness.
The regular meals, sometimes you gotta just force yourself to do it, but it ends up going right every time.
Yeah, I mean, the food on the ship has been incredible.
Incredible.
Like, I definitely don't eat this well at home.
No way.
I'm going to be really sad when I leave and have to cook meals for myself.
I don't bake nearly as well as the guys here.
Yeah, same.
I know, whatever they were baking for the, like, they give us like an afternoon snack, and whatever they were baking for it smelled so good. It was like coming through the
Oh
vents
my
on the way
god.
up here.
I can't wait.
So everyone get excited for that once our shift's over.
Yesterday's was like so good. I don't know what
I
it was
didn't
like.
get any of it.
It was like a little like cookie type pastry, like really flaky, and then it had like some jam and maybe like some type of nut on top. It was really really good. I had like 2 of them.
Yeah, the quality of the food is one thing, like it's so absolutely fantastic, but I'm also always impressed by the like options available. Like over the course of a month or two, I'm sure it gets a little more familiar, but it's just nice. There's like rice and pasta and quinoa and then a few different types of meats and a few different types of veggies. And I don't know, I've just been so totally impressed by it.
Same. It's great. Like they accommodate for all of our like dietary restrictions and things like that. There's always tons of options to choose from. It's almost like a little buffet. We have fresh fruit every day, fresh veggies every day. Amazing coffee. We even have a nice espresso machine that we get to use from time to time. Very, very delicious.
Very nice, yeah. The buffet spread has been top notch.
I had an experience with the espresso machine where, so apparently the like thing to steam the milk is supposed to have like a pouch of milk attached to it. But I didn't know this. So I was like, okay, I'll try this out. I'll try like a macchiato or whatever it does. And I press the button and then it just started shooting steam everywhere.
Okay, I'm glad you did that, not me, because I was about to. I haven't tried it yet. I didn't know if it worked, but good to know.
Yeah, jot that down. So you can do like the espresso and the coffee, but you can't do anything that involves steam milk.
Gotcha.
Because the milk pouch has gone missing.
Oh no. Definitely good to know. No steam milk in the future. Do y'all know who dropped off these delicious, cute little pineapple treats? Megan? Okay, they're so adorable. I love them so much. Cool. Oh, we're getting closer to the bottom. So exciting.
Yeah, shout out to Alex and Alexander and Jose, who are our cooks and steward on this trip. They're doing a great job. We love you.
Yes, definitely a big shout out to our cooks. They are amazing. They're probably the hardest workers on the ship, to be honest.
Yeah, huge shout out to the crew in general. They've just been so incredible. And they're all, everyone on the ship is like so nice and friendly. And it's so nice to chat with everyone.
They're great. I love them. Last night, I don't know if I told you this, Leela, but last night I was up just kind of getting used to the 12 to 4 shift. Even though we weren't doing a dive, I stayed up for my 12 or my midnight to 4 a.m. shift. And to kind of keep myself awake, I had my headphones in and was having a little dance party on the social deck by myself. And I turned around and a couple of the crew members were just looking at me laughing. I was like, what? Come join, come join. They were like, no, you're good, you're good.
Could do one of those like raves where everyone wears the silent raves,
Yes.
everyone wears their own set of headphones. Well, sounds like you were the entertainment for the evening.
Definitely. I entertained myself. I'm glad I could entertain someone else too.
What were you listening to?
Oh gosh. Like, I was definitely listening to some Gaga. She's my queen.
That was actually my guess. Because like, if there's music you really have to like get up and dance to.
Oh yeah,
it's
definitely.
Gaga.
For sure, for sure. check some questions here. Oh, this one looks interesting. So, I wonder, Steve, maybe you can help me answer this a little bit. We have a question about maybe what kind of species would we expect to see on this expedition or at these depths as we're heading down to around 2,200 meters.
Yeah, so we're diving on seamounts, and seamounts are basically extinct underwater volcanoes, and they're full of rocky, hard substrates that a lot of things that can attach to surfaces like to attach to, so corals and sponges. Coincidentally, seamounts are really great for high-speed current environments, as currents are topographically accelerated over shapes on the seafloor. So a lot of the things that we're going to see are probably going to be filter feeders and suspension feeders, but there's also groups of animals that will feed on the deep sea sediments, deposit feeders, things like maybe sea stars or urchins or sea cucumbers. Occasionally we'll see fishes, but I don't expect fishes to be a huge part of the community here. But
Gotcha.
especially deeper, you don't see them as much.
Okay.
But it's possible.
Does that have to do with like the pressure down there or the temperature?
It's mostly food limitations.
Okay.
So a lot of the things that if we do see fishes, they'll probably be things like rat tails or grenadier fish. Occasionally you'll see small midwater fishes will drift down. We saw a few lantern fishes on our way down here through the deep scattering there. Occasionally you'll happen upon a deepwater shark, but I think we're going to be a little bit deep for that on this dive. We'll almost certainly see halosaurs.
Oh, so cool.
Halosaur fishes, and possibly even some eels. Some cutthroat eels are pretty common in these types of features.
Very cool. Yeah, that's something I recently learned about fish, and you said something about sharks. I recently learned that most of the shark species, most shark species actually don't live in shallower waters, like what we think of Jaws and the tiger sharks and the great whites. The majority of the species live in deeper waters, which I thought was super interesting.
Mm-hmm. I'm just going to nod and say yes, I'm not a fish. So sounds good to me. That's why we have a science chat. We have people who are fish experts joining us and able to convey their expertise. We can't bring everyone along with us, but we have near limitless expertise on shore.
Yeah, that's great that we're able to work with all these scientists from around the world on shore. Experts in all sorts of different fields and different species. So, Mariana, you are from the Republic of the Marshall Islands. Could you remind me again which island you're from?
From Majuro or
Oh, okay. Awesome.
I'm from Island Lovelock too.
Okay,
Yeah,
gotcha.
so around here.
So what's it like to be on this experience and get to explore these waters? I'm sure it has some pretty significant meaning to get to go out here and explore these waters of your home.
Do you hear? Can you hear me? It's very cool. It's very, to me it's like a dream come true.
Yeah.
Actually, cause I never I never imagined being in here
Definitely.
and participating in this expedition, cause you know we're from a very small country, island country, so
Right. Yeah,
yeah.
we're so thrilled to have you on board and learn a little bit more about the Marshall Islands and a little bit more about the culture. And I was just wondering, do any of these waters have like any significance in Marshallese culture, or were they like used in the past for any significant reasons or
Oh
anything like
yeah,
that?
you know the Marshall Islands. There's like more land, more water from land.
Uh huh.
So we pretty much respect the water and we take care of the water and our resources. It's our only resource.
Yeah,
Basically.
that's
Yeah.
great. I
So.
love that. I love that you guys, your culture kind of embodies
Mm,
that respect of the water and the ocean and the organisms that live there. I think that's
yeah.
super important.
How do I? How do I?
So, Steve, I know you said that a lot of the species that we may encounter are kind of like filter feeders and just kind of collect stuff that flows through the water column with the current. Are they what are they like collecting? What are they eating? Is it just like smaller organisms or like pieces of organisms?
Yeah, both of those things.
Okay.
Sometimes it's whole organisms. Uh, Most of the stuff that, everything falls to the seafloor in one way or the other. And so one of the ways we categorize stuff in the water column is, it's called marine snow. So this is just flocculate material that sticks together because it's organic and and mucusy maybe, and that stuff is a food source because it contains useful carbon that animals can intake. But sometimes you'll see, especially near bottom, whole organisms can be captured out of the water column by other animals.
Okay.
Small crustaceans even. Some things will actually try to capture whole fishes.
Oh wow.
It depends on the depth, but depends on the depth where we're at. And though the deeper we go, the tendency tends to be more passive filter and suspension feeding. Yep.
Very cool. Yeah, that marine snow, I've heard a little bit about that. I can't remember. Is it the vampire octopus or squid that feeds on it? It's one of the. Do you remember?
There's a vampire squid, yeah.
Okay.
Yep. They eat yeah, marine snow and sometimes small animals.
Very cool.
There's other groups of animals that are scavengers. For example, they'll eat whatever falls to the bottom. As you might imagine, whales, fish, all these things eventually sink to the seafloor, and they're pretty bountiful
Yeah.
feasts for anything that might feed on the seafloor.
Yeah, for those of you listening, if you haven't, I highly recommend checking out the gallery on nautiluslive.org. There is a video of a whale fall that was discovered on a previous expedition, and like Steve said, it's just like a smorgasbord. All the organisms come in to feed off of this food source that has fallen down to the bottom of the ocean. So those of you who are just joining on our live feed on nautiluslive.org or maybe even on YouTube, we are on our first dive of NA174 as we head down to the depths of the ocean right around 2,200 meters on a very newly mapped seamount. We actually mapped it just last night as we are arriving in this location, and one of the main reasons we like to do that mapping first, it gives us basically like a 3D image of what the seafloor looks like. So just like on land, the seafloor has lots of cool geologic and topographic features. There's valleys, there's ridges, mountains, canyons, and so we kind of use that mapping data to help us pinpoint where we want to do our ROV dives. And again, those of you who are looking to get more information on that, the well fall that I just mentioned, again, you can go to nautiluslive.org, and then in the top right, you can click on Gallery, and you will see featured topics. And one of the very first ones is well falls, and it has some really amazing footage of some of the well falls that OET has seen throughout previous expeditions. Getting closer. Just over 2,000 meters in depth as we approach that seamount. Again, heading down to about 2,200 meters. Annette or Steve, I just had a question come in about how many dives we might be doing on this specific expedition. I know that it really depends on a multitude of factors, but do we have an estimate of how many dives we might be doing on this specific expedition, NA174?
As many as possible.
Yeah, definitely. I like to hear that.
It's tough to tell because we have to map most of the sites that we're going to dive on. So depending on how long that might take, would significantly affect how many dives we
Gotcha.
could do.
Yeah, definitely makes sense. Yeah, we definitely want to do those maps first so we can get a better idea of what we're diving down into. Again, getting closer to our target depth, around 2,200 meters on this dive. We're right about 2,100 meters. The current temperature for Hercules is about 2.1 degrees Celsius with right around 30% oxygen saturation. So these little white specks that we're seeing on the screen, that would be marine snow probably?
Yeah.
Okay.
Probably. Yeah, they tend to zoom by when we're going down. It's hard to tell, but there's some small organisms in there too.
Yeah, yeah. Some of them look like they're controlling their movement. And yes, as we map these areas, like I said earlier, it kind of gives us a 3D image of what the seafloor looks like, and we can see these really amazing geological and topographic features, and kind of pinpoint where we want to do our dives based on where we might find more biodiverse areas, or where we might find some of the different rocks for our geological samples that we're collecting. Really just help us pinpoint the location that's best going to allow us to meet our overall objectives. Hey, Gabby, would you have a second to answer a question? Would now be a good time?
Yeah, sure thing.
So we had a question about, specifically for you, like what is the role of the navigator as we're doing these dives?
Yeah, so navigator has to maintain like a awareness of not just the ROVs underwater, but the boat itself. So our job is to communicate with the bridge,
Okay,
and
gotcha.
then also communicate with everybody here, the pilot and of Herc and Atalanta. So if we need to move the ROVs, I also have to move the boat.
Gotcha,
Yeah.
okay.
So at the same time, I have to know the currents, the winds outside, if it's pushing the boat. Yeah.
Yeah, lots of knowledge, lots of things go into play there, I'm sure, definitely.
Yes.
So we've made it to the seafloor as we're exploring this really cool seamount. So exciting.
So I'm going to turn on multipuffer samplers 1, 0, and 1.
0 and 1.
Yes. They should be switching on now. 0
Okay.
and 1. Looks like we have flow. Good?
Yes, plugging them in now.
Okay.
And then confirming it's pump 0 and pump 1.
Yes.
Okay.
So
Do I have to take
exciting.
an in situ?
So exciting. For those of you that just
And then
joined
do I
us.
hit a second one for, okay. Sorry.
No, you're fine. For those of you that are just joining us, we just made it to the seafloor, right around 2,200 meters in depth on our first dive of NA174, just east of Isling Laplap in the waters of the Republic of the Marshall Islands. And so what you're seeing through these cameras is coming from a remotely operated vehicle. So this is not a submarine. This is an unmanned vehicle. We are actually on EV Nautilus right now. And we have pilots that are basically driving these underwater vehicles as we explore the ocean floor. Yay, I'm so excited we're on the seafloor. So what we're seeing here is you can see all these rocks. This may be part of a collapse, some debris from a previous collapse of like a larger seamount or rock or something possibly. Just coming up from higher. Okay, gotcha. So yeah, so some sort of collapse, debris of these rocks falling down to the seafloor, which may be some really cool specimens for us to collect for our geologic analysis. We have another question that just came in about our ROVs. Is one of them just kind of like support for overall viewing while the others are up close? So for those of you that are just joining us, we actually have 2 ROVs in the water right now. They work in kind of a dual body system. The one that is closer to the surface is ROV Atalanta, and then at the bottom we have ROV Hercules, and they work in tandem. As you can see in our view, we have ROV Hercules has these arms that are capable of moving around and grabbing and picking up different samples. So we may be able to use them to collect some of our rocks for our geologic analysis.
Let us know when you're situated, and we're gonna, first order of business is pick up a rock, if we can find one. Yeah, and if we don't find anything, we'll move on. But can we get lasers on too? Yeah. Anything over here? Or anything that...
There's a little red shrimpy friend.
Uh, not promising, but we're willing to poke at a couple things if we see anything pretty obvious. But yeah, let's take a look at in this next pile. If we don't see anything there, we'll put in a move and move up a bit. Yeah. Well, we landed in a flatter area, so I'm hoping as we move up slope a bit, there might be some more broken material. So I'd keep an eye out. But I haven't seen any large animals yet, some small crustaceans around. I think the most interesting thing on this site that I've seen so far, actually, there's a small crustacean-y thing there. Oh. Telestrator.
Very cool. And for those of you watching, those lasers are basically allow us to determine the size. They are 10 centimeters apart.
That's probably better. Usually it's just, it just gets minimized. There it is. So can we zoom in on this thing? Yeah, it's a cucumber.
Awesome.
All right. We'll just get some captures of that and move on.
Any idea what species or?
Very difficult.
Yeah.
If I had to put it in a genus, it's probably Peniagone or Amperima. They're two that look very similar.
Very cool. I love its body's like see-through.
All right, let's resume the search for a rock, and if we don't see anything in the immediate area, we'll put in a move.
Very good eye, Steve.
The one thing that I'm most excited about at this site is actually the ripples in the sediment so far. And that kind of is a indicator of something that kind of drove my fascination with this particular ridge, is that if you look at the orientation of the ripples, it's actually north-south across the slope.
Okay.
And so that indicates that there's some persistent periodic north north or northeast deep water current that's causing ripples in the sediment. This is different from the shallow water waves. These are
Gotcha.
produced almost exclusively, probably at interfaces and water masses.
Oh, cool. Is that a good indicator of life, like that we might find something living there?
That one's a bit big. What do you think about that? No, no. No, I think we're going to pass on that one. But that's the right size.
Looks like we have a little crustacean down there, maybe?
Oh, there's a small pigurid.
Gotcha.
Common name, hermit crab.
That's what I thought. Yeah. Okay.
Yeah.
And then is that another little crustacean, the red one?
Yep. Yeah,
Awesome.
we'll see a lot of small shrimps and things.
Very
like
cool.
That. Any of these? Yeah, how about this one over here? What do you think about that? You see the telestrator up there front row? Or this one? Well, this one I think is better.
It almost looks like it's kind of sitting on top of another rock.
Yeah.
I love your little illustrator. It's so neat.
Looks pretty stuck. All right.
Also, before we poke anything, just give me a heads up so I can get a sample in situ photo first.
And so we're wanting to collect these rocks for geological analysis. They can tell us a little bit more about the history of life on Earth as we can analyze them and try and determine their age. Is that right?
Yeah, I think these are going to be in place. I think we can keep looking.
Maybe a little friend floating by there too.
Yeah. Okay. So we're not gonna poke it.
Okay. I think we can move forward. No, we'll just go perpendicular upslope towards to or parallel to it. There's nothing magical about the points. You don't have to thread the needle. You can just kind of go perpendicular upslope in that general direction.
Steve, when you say in that general direction?
Towards waypoint 2.
Cool,
yeah.
Thank you.
300 or 30.
They're more of what we call guidelines.
Should this... Oh.
But I can change my heading and you can swing out. Is that towards the waypoint or?
Yeah, that looks good.
Sure. Nice. happening? Are you referencing Shrek?
I love that movie.
We're gonna get along great, Levi.
This is
Bubble, where art thou? I'm gonna cycle bubble cam. Oh no, never mind. I got him back.
So the port manip is in the field of view of the wide field camera. Is there anything you can do about that?
Absolutely. Do you get if I move that around? Levi? Okay. Someone is on their mic buzzing around. Okay. Let me know which one you're looking at. This slower one? I'll also pitch out a little bit. Is that out of your way? Which camera was I worried about? Whoever was requesting camera stuff?
You mean that? Yeah, we can take a quick look. Oh, it's a sea cucumber.
Cool.
It's probably the same species we just saw, but they do have the ability to swim. So this one is the same, probably Peniagone or Amperima.
Wow. Is that like an internal organ that we can see inside of it?
That's the gut, yeah.
So cool.
It's full of sediment.
Steve, what were the two potential species?
Peniagone or Amperima. I can put them in the chat.
That would be great.
Yeah.
Thank you.
That's awesome.
Actually, I'll just put them in the event log.
Cool.
Wow.
Yeah, you're getting pretty.
And so is that one of those filter feeding species?
It's a deposit feeder.
Okay.
So the tentacles are on the upper left side, and it uses those tentacles to move sediment towards its mouth.
Gotcha. So cool.
Right.
What about those little appendages on the bottom? They like have white tips.
Yeah, that's it's part of the swimming apparatus.
Okay.
Yeah, I don't have a name for it, but they typically have appendages that are more flare that allows them to give a little bit of
Gotcha.
surface area to push water away with.
Totally. Such a cool adaptation. So neat.
Yeah, I think that's good enough.
Steve?
Yeah.
I think you were the one asking for port arm to move?
Yes.
Is that...
It is
was
looking
that in
good.
lower cam that you were? Yeah, okay.
Yeah, it looks good now.
Perfect.
Yep.
Roger.
How are we doing on the move? Are we still moving? Okay. Yeah, I think I think we're at a good time because we're approaching the watch change, and if there's something maybe in this area that looks promising, maybe we can set down and do a little bit of sampling, and it'll transition us into watch change. Like maybe
what exactly
this one?
are we looking for? Okay, loose rocks. Possibly one of those quite small ones? I mean, we haven't poked anything yet, so like...
Yeah, I think... yeah, It's tough to tell with these, but if there's something that looks like you can get a purchase on it, I think I'm looking at this one right here. But there's a lot of good candidates here. But it's tough to tell until you get
I got you.
close up in on it.
You want to see your porch? There you go.
Can we poke this one? I think
Let's do it.
it looks
Let
like a candidate.
me-
You might be able to get one of your jaws
Give it a
under.
poke.
ROV, let me get a photo of it real quick. Steve,
Yep.
could you
Yep.
circle that one more time?
Yeah, the lasers
okay,
are
it's
on
that
it right now.
All right, all good. Go ahead.
I'm pausing the samplers while we do this.
Noted.
Yeah. Do you want me to poke it before we do the changeout? Okay. We're getting a situ picture of it.
We good on images?
Yes,
Yeah.
all good.
Good
Thank
on
you.
Images? Okay. What? It's just not reading. It hasn't been reading for a while. I'm just gonna poke this rock and then you can take over. Okay. All right.
I turned.
Talking about. All right, we're talking about this one. Ooh, You talking about this one, Steve?
Yeah. Can you get a jaw finger under one side of that and see if
Yeah, sorry, Levi.
it's pretty solid?
That's pretty in there.
Okay.
Yeah.
All right, well, that's about the right size, but
I'm gonna poke.
yeah, we
like
want something
this one.
free, so
Yeah. I don't know. Here, let me just try to. No, that's nothing.
that's called a mountain.
Yeah, that's part of the. It's part of the landscape here.
Yeah, yeah. All right, well, yeah, I think that was our best option here. I think we'll keep looking.
Okay, Roger. All right, we're doing changeover.
All right.